Let us start the discussion about the one of the important topics of molecular basis of inheritance, that is the search for genetic material. Search for genetic material. Actually, there are different types of the experiments were performed to identify the genetic material in living organisms, and the first experiments which is performed to identify the genetic material in living organism is called as the Griffith experiment. The first experiment which is conducted to identify the genetic material in living organism is called as the Griffith experiment, and this Griffith experiment is based on the phenomena of bacterial transformation. The Griffith experiment is based on the phenomena of bacterial transformation. So first of all, here we discuss what is the meaning of the transformation. First of all, here we discuss the meaning of the transformation. Suppose, for example, two bacteria is there. Here is a one bacteria called as the A, and here is a another bacteria called as a B. If the genetic material is transferred from one bacterium to another bacterium, if the genetic material is transferred from one bacterium to another bacterium with the help of or through the liquid medium, is called as the transformation. So what is the meaning of the transformation? I am repeat again. Transformation, simply it is a process of the transfer of the genetic material from one bacterium to another bacterium through the liquid medium is called as the transformation. Second point, there is one bacterial species called as the Streptococcus pneumonia. There is one bacterial species called as the Streptococcus pneumonia, and this Streptococcus pneumonia these bacteria undergo transformation naturally. The bacterial species called as the Streptococcus pneumonia, these bacteria undergo transformation naturally. So the scientist Frederick Griffith used the bacterial species known as the Streptococcus pneumonia for his experimental work. The pathogenic species called as the Streptococcus pneumonia, actually it had two kinds of the strains. The bacterial species called as the Streptococcus pneumonia, actually it had the two kinds of the strain. What is the meaning of the strain? Simply, strain stands for the type of bacteria. Strain, the word strain stands for the type of the bacteria. There are two different kinds of the strains belonging to the species Streptococcus pneumonia. First strain is the S strain. First type of the strain is called as the S strain. On the other hand, the second kinds of the strain is called as the R strain. These two strains used by the Frederick Griffith scientist for his research work, that is the S strain, and second type is the R strain. So what is the meaning of the S strain? What is the meaning of the S strain? Suppose, for example, here there is one bacterium, and if the bacterium show the presence of a layer around a cell wall which is made up of the polysaccharides, and this layer is called as the capsule. What is the meaning of the capsule? Simply capsule, it is a protective covering around the bacterial cell wall known as the capsule. These strain A strain show the presence of the capsule, therefore the appearance of the S strain is smooth. Therefore the appearance of this strain is smooth and called as the S strain. On the other hand, the R strain do not show the presence of the capsule. R strain do not show the presence of the capsule, and therefore the appearance of these kinds of the strain is the rough. Therefore the appearance of these kinds of the strain is rough, called as the R strain. And we know that actually the pathogenic properties of these bacterial species is due to the presence of these capsules. Pathogenic property or immunogenic property is only due to the presence of the capsules. And second important point regarding capsule is that the capsule, it is a layer which protect the bacterium. Capsule, it is a layer which protect the bacterium from host immune system. Capsule, it is a layer which protect the bacterium from host immune system. And the third point regarding capsule is that capsule, this layer is made up of polysaccharides. Capsule, this layer is made up of polysaccharide. The fourth point regarding capsule is that the formation of the capsule is under genetic control. The formation of the capsule is under genetic control. We elaborated this statement, that is the formation of the capsule is under genetic control. How exactly the capsule is formed under genetic control? For example, here S strain is there, and this is the genome of the S strain, and the genome show the presence of specialized genes called as the capsule encoding genes. The genome of the S strain show the presence of specialized gene known as the capsule encoding genes, and these capsule encoding genes express to form the various enzymes, and these enzymes are actually responsible for the formation of the polysaccharide layer. These enzymes are actually responsible for the formation of the polysaccharide layer. Okay, so due to the presence of the capsule, these kinds of the strain is pathogenic in nature. Due to the presence of the capsule, these kinds of the strain is pathogenic in nature. On the other hand, the R strain do not show the presence of the capsule. R strain do not show the presence of the capsule. Here it is therefore it is a non-pathogenic strain. Therefore it is a non-pathogenic strain. Then next distinguishing features of the S strain is that S strain is the virulent strain which cause the pneumonia in mammals. On the other hand, the R strain is non-virulent strain. R strain is non-virulent strain which do not cause the pneumonia. So I am repeat again. The Griffith experiment as usual is performed by a medical officer, Frederick Griffith, and for his research work he used the bacterial species called as the Streptococcus pneumonia. And in Streptococcus pneumonia, two strain is there. Strain simply means the type of bacteria. First strain is the S strain. On the other hand, the second strain is the R strain. The S strain show the presence of the capsule around the cell wall. Therefore, the appearance of these kinds of the strain is smooth. On the other hand, the second strains called as the R strain do not show the presence of the capsule. Therefore, their appearance is the rough. Then, second points, this S strain is pathogenic in nature due to the presence of the capsule. On the other hand, R strain is non-pathogenic in nature due to the absence of the capsule. Then next point, A strain is the virulent types of the strain, while R strain is the non-virulent type of the strain. Then we discuss some of the important characteristic features regarding the capsule. First point, capsule has the immunogenic properties. Second point, regarding the function of the capsule protect the bacterium from the host immune system. And third point, capsule, it is a layer of the polysaccharides. And fourth point is the formation of the capsule is under genetic control. Here we elaborated this. The S strain show the presence of capsule encoding gene in their genome. These capsule encoding genes express to form the various enzymes, and these enzymes is responsible to form a coat of polysaccharide which is known as the capsule. Now let us start the discussion about the steps of Griffith experiment. Let us start the discussion about the steps of Griffith experiment. In the first step, Griffith injected A strain into mice. He make the liquid nutrient culture medium, and in liquid nutrient culture medium he added the A strain, and now the A strain is transferred into the mice. As we know that this A strain show the presence of the capsule, and because of the capsule, A strain is the pathogenic. Therefore, mice dead. Death of the mice takes place in this step. In second step, the scientist Griffith injected R strain of the bacteria into mice, and as we know that the R strain is non-pathogenic. Door laun ke bara. R strain is non-pathogenic, and due to in this step mice alive. Then in the third step, Griffith killed A strain with the help of the heat. Now it is called as the heat killed A strain. Now it is called as the heat killed A strain, and he introduced heat kill A strain into mice. But as we know that in this step the A strain killed with the help of the heat, and the capsule layer destroyed due to the treatment of the heat, and therefore mice unaffected. death of the mice does not takes place in these steps. Now in the fourth step, Griffith take the mixture of heat killed strain. He prepare the mixture of the heat killed strain as well as the R strain, and with the help of the ejection he transfer this mixture into mice, and surprisingly he found that death of the mice takes place in fourth steps. So here just revise the steps of the Griffith experiment. In the first step, Griffith took the culture of the S strain and transferred into mice with the help of injection. As we know that the S strain is the pathogenic, therefore the death of the mice takes place during the first step. Now in second step he injected the R strain into mice, but as we know that the R strain is non-pathogenic in nature. R strain is non-pathogenic in nature, therefore mice alive during this step. And in the third step he injected heat killed S strain into mice, and in this step mice alive due to because of the rupture of the capsules. And in the fourth step he take the mixture of the heat killed S strain as well as the R strain and transfer this mixture into mice, and therefore the death of the mice takes place in the fourth step. Griffith surprisingly found that the mice dead in the fourth step. Okay, so therefore he collected the blood from these dead mice. He collected the blood from these dead mice and grow on a culture medium. He collected the blood from fourth step and grow on culture medium, and he surprisingly found that the culture medium contain only one kinds of the strain, that is nothing but the S strain. In this step actually Griffith used two kinds of the strain. He used heat killed S strain as well as the R strain. But when he cultured the blood from these dead mice on a nutrient medium, then he found that this culture medium contain only one kinds of the strain called as the S strain. So let us start the discussion about the interpretation. Result. How exactly Griffith found only one kinds of the strain, that is the S strain? Right now here we discussed in details. For example, here it is a heat killed S strain. Here it is heat killed S strain, and here it is a R strain. Here it is a heat kill A strain and here it is a R strain. Now there is a specific transforming principle. There is a specific substance. He called it as transforming principle. Now the transforming principle is transferred from heat kill A strain and from heat kill A strain into R strain, and the expression of this transforming principle takes place, and due to the expression of the transforming principle, a layer of polysaccharide is synthesized around the cell wall, and therefore the R strain is changed into S strain. Therefore the R strain is changed into the S strain. Today we know exactly what is this transforming principle, but Griffith do not conclude the exact transforming principle. What is the chemical nature of the transforming principle? Whether this transforming principle is DNA, whether this transforming principle is RNA, or whether this transforming principle is protein. Griffith do not know much about this. So the drawback of the Griffith experiment is that Griffith is not aware about the chemical nature of transforming principle. Griffith is not aware about the chemical nature of the transforming principle, and therefore further experiment is performed by the three scientists Avery, MacLeod and McCarthy. So in this manner, in detail we discuss about the Griffith experiment, the steps of the Griffith experiment, as well as the interpretation of this result. Thank you.